Hey guys, moving on to transcription. We've got, a couple more weeks, probably a little bit more maybe, because I got a couple activities that I'm working up to get through this unit. We'll have a little test, actually probably more like 3 or 4 weeks. And then we'll have the last couple of weeks to really get prepared for the AP exam. So no worries. We'll go through how the exam is going to be modified. We'll go through what to look out for. And we'll give you strategies and everything for that exam when we get closer to it. Right now, though, we need to finish up this unit. So we've got a while still. We've got to get through transcription, translation, genetic technology, gene expression. There's a lot of other things we still need to do. Mutation, we still have to do that as well. So there's a lot of stuff we got to get through with this. So we'll get through this. You'll be prepared. Don't worry about it. If you're looking at the AP website and going, oh my gosh, what are we going to do about this? We'll take care of all of that. We'll sort it. Okay. No worries. You'll be fine. You'll do well. I have complete confidence in you guys. All right. Apparently we're having a cat with us again today. This is Teeny. Hi, Teeny. Bye, Teeny. She's not really social. Okay. So getting from a gene to a protein, that's a process called protein synthesis. The first step of protein synthesis, of course, is transcription. The second step is translation, but we care about transcription right now. And I did make the sound effects go away because I realized they were coming through on the video terribly. So you should do this without sound from here on out. So the DNA, of course, codes for who you are. You've heard me say it's the recipe for who you are. And if you're a cat or, a fish or a goat or whatever, a bread mold, what you are is determined by what those genes are, and they are recipes for proteins. So we use DNA to make proteins. And of course, proteins are where we get our traits, because proteins are what tell our cells. Okay, so the sound's not gone. Hopefully you didn't hear that. Maybe you did. The proteins you find in a cell determine what the cell's function is, how it works, what it does. And then, of course, those cells working together, that's what makes up us. So the flow of genetic information in a cell, like I said, more specifically, we've said DNA replication is part of that. We've been through that. DNA gets transcribed into RNA and then gets translated into proteins. So that's transcription and that's translation. And those proteins, of course, like I said, cells, bodies, traits, right? That's what makes us who we are. And this is Francis Crick up here at the top right. Of course, he's the one that coined the phrase central dogma, and he did apologize for that later on. He said when he coined the phrase, he didn't know what the word dogma meant, and later when he found out that it means handed down from God and should not be questioned, he realized he probably shouldn't have called it that since he was the one presenting the idea. So we learned that DNA is the source of our traits through metabolic processes. We realize that it's a failure to metabolize things properly that makes us have some genetic disorder. So it's a failure of a protein. And so once we determine that DNA is the code for proteins through Nuremberg's experiments and many others, we started making this connection. We started seeing that you can get the same disease, right? One of the things we learned is that you can get the same disease a number of different ways. If you have a problem, right, with enzyme 1 or enzyme 2 or enzyme 3 or enzyme 4, either way you end up with the same disease because you have to have whatever E is on this little diagram. You have to have that to not have the disease. So there are different ways to get the same disease. And oftentimes these enzymes will catalyze other reactions and diseases may have more complex sets of symptoms. So when we're looking at transcription, we're going to take the code that's in DNA and transcribe it into that same code in RNA. And we're going to make some adjustments, some tweaks to that strand of RNA. So it won't be the exact same code as what was in the DNA, but you'll see what those changes are and why we make them. So first of all, we need to talk about the differences between RNA and DNA. Why don't you see if you can think of any? You may remember some. So differences between RNA and DNA. Pause the video and try and come up with 3 or 4. So hopefully you remember that RNA normally comes in a single strand, whereas DNA is that double helix. Really the whole idea of DNA, right? RNAs are really the molecules that do stuff in the cell. And DNA is really just like a safe and stable way to save the codes for the RNA. We often think of DNA as the central idea, the main molecule here, but in reality, it's just a more permanent way to save our RNA codes. What we need are the RNA codes. That's the thing we're going to do something with. DNA is just a safe way and stable way to keep those. The whole idea of the nucleus, the whole idea of being a double helix where the code is, the sequence of bases is protected by the outer part of that molecule as it twists up. Now, having said that, there are, of course, changes that occur and we get mutations, and we'll talk about those later. But so RNA is single-stranded, DNA is double-stranded, a double helix, anti-parallel. The sugar in RNA is ribose. The sugar in DNA is deoxyribose. You probably remember that RNA uses uracil instead of thymine, a TCG for DNA, and adenine pairs with uracil, with A goes with U instead of with T in RNA. Good. Some different types of RNA. There's lots of different types. You probably remember messenger RNA, which we'll generally call a transcript. The product of transcription is messenger RNA. But of course, there's ribosomal RNA. Part of the ribosome is made up of RNA. There's transfer RNA, tRNA, which we'll talk about when we do translation. But there's lots of other RNAs. There's microRNAs, there's long non-coding RNAs, there's SNRP RNA, there's telomerase RNA. There's a lot of different RNAs out there. So what determines if a gene is going to be transcribed? And we'll go into a lot more detail with this later when we talk about gene expression. You're not going to just transcribe any old stretch of DNA. There are specific genes that you need to code for. And the part of your genome that codes for messenger RNA is only about 1.5% of your genome. It's not most of it. These genes are rare. So they have to be clearly marked. They have what's called a promoter sequence. And a promoter sequence is the little sequence of DNA. It's a very specific set of base pairs that basically says, here's a gene. Specific transcription factors, proteins, bind to specific promoter sequences. So that promoter, though, is what says, here's the gene we're going to transcribe. So if the transcription factors are bound to the promoter in the proper way, then it will be transcribed. There can also be transcription factors that shut off genes. They will bind in a way that blocks transcription. The actual protein complex that does transcription is called RNA polymerase, and it is a big complex of proteins. And it's got a good, simple name, RNA polymerase, polymer, putting many units together. ASE at the end, it's an enzyme, and RNA is what it's putting together. It makes a polymer of RNA nucleotides. And in general terms, basically all we're going to do is we're going to unzip the DNA. We're going to use one of the strands to copy. We're going to assemble an RNA strand. This is a lot like what we did with DNA, where we unzipped and built two new strands. But in this case, we're only going to build off of one side. And on a chromosome, it doesn't matter which side. There may be a gene on this strand at one location, and you go farther down the chromosome, and the gene's on the other strand. It's completely arbitrary. So we unzip, we build a complementary strand that's made of RNA, has U's instead of T's, uses ribose instead of deoxyribose, and then that can peel off and go become our messenger RNA. And then we'll zip the gene back together when we're done doing transcription. RNA, of course, is a nucleic acid, and nucleic acids are always built 5' to 3'. We extend the 3' end, so the 5' end is there. So when we unzip the DNA, the strand of DNA we're going to be looking at would have 3' this end and 5' this way. It would be running this way, and we're going to build a strand running this way, anti-parallel to it, just like the base pairing. Following the regular base pair rules, of course, it has to be anti-parallel. So we're going to build 5' to 3'. That's the way we always build. So here's a little diagram, shows how we're going to unzip this, which just shows RNA polymerase. There's a big pink wad of bubble gum in the back here, but we are unwinding and unzipping the DNA and then rewinding and rezipping the DNA behind it and just opening up a little bubble, transcribing as we go and closing it as we go and generating this mRNA. And again, we are extending the 3' end. So the next one on here is going to be an A, and the next one after that's going to be a U because that's a T and that's an A. We're just going to keep adding to that end as we continue to head in this direction. So we have two different strands of the DNA. We have a template strand, and the template strand is the one we're actually building off of. So the template strand if it says a T, I'm going to stick an A of RNA on there. If it says G, I'm going to put a C of RNA next, right? So that's my template. Now, the other strand that we're not using for this particular gene, that's called the coding strand. Its code is going to be basically the same as the RNA we're building, only, of course, it's going to have T's and it's going to have deoxyribose, blah, blah, blah. But that's called the coding strand. The one you really need to know about is the template strand. That's the one that gets asked about. The template strand is the one we use base pair rules to build the RNA strand. The template strand is also considered antisense. The messenger RNA is sense. So the coding strand is sense, and the template strand is antisense. It's the opposite. It has C's where the coding strand would have G's. So looking again at this diagram, we have the coding strand, we have the template strand. The coding strand is sense because it's the same sequence. Look, I got a T, a G, a C, a C, a T, a U, an A, a C, a C. It's the same sequence as the mRNA, and the mRNA is considered sense. And the template strand is antisense. It's just the opposite. Pretty straightforward. You need to make sure those terms make sense to you. So again, looking at which genes are going to be transcribed, it's all about the transcription factors binding to the promoter sequence. So the appropriate transcription factors that say, yeah, let's make this one, what they're going to do is they're going to summon RNA polymerase. They're going to be attractive to RNA polymerase. And so as these bind to the DNA and then they attract RNA polymerase can take off and start transcribing this gene. But it's all about the transcription factors binding to that promoter sequence. So we have this promoter sequence, which is still considered part of the gene, but it's not actually going to be transcribed. It's just the place where the transcription factors bind. Then we have the RNA polymerase is going to be called in right here, and it's going to be able to work this direction off of that, building off of that template strand. The transcription factors and the RNA polymerase all together, sometimes that's called a transcription initiation complex. It's just words that mean we're going to start doing transcription here with these big molecules. Eukaryotic genes are not continuous, which means that you don't just start building RNA and that RNA is going to be exactly what we use. We're actually going to use a lot less. The stretch of DNA has parts in it that are not necessary for the protein, for the mRNA. And so those parts have to be cut out. So those parts are called introns, and introns are going to be removed. So we're going to take introns out of that mRNA. The DNA stays intact, right? But it unzips, it builds the RNA, and as it's building it, it snips out little pieces. And those little pieces are called introns. What remains are called exons. I've never heard anybody ask really about exons, but introns get asked about all the time. So we have introns. Eukaryotic organisms have introns in their genes. It's just extra code that is not part of the actual recipe. And we'll talk more about them later because there are some kind of interesting and complicated things that happen with introns. But for now, we just need to understand that those introns have to be removed. And we call that mRNA splicing or transcript splicing or transcript editing, different terms that all mean basically taking out the introns. So a mature transcript has the introns removed. And we have to splice it just right. If you take out the wrong part or take one nucleotide too many, you're going to screw up everything after that. So we'll talk more about that when we do mutation. So the part that removes introns, they're called SNRPs, and they are small nuclear RNA protein complexes. So they're small, they're nuclear, they're in the nucleus, they're made up of RNA, and they're made up of proteins. And they remove introns, and they're called SNRPs. Yeah, legacy. My response. Such respect. When the Human Genome Project was first started, we expected there to be over 100,000 genes. And the reason for that is because there were over 100,000 proteins. We still don't really know a good number of how many proteins we use in our bodies. It's a lot. But what's interesting is that we only have about 21,000 genes. So we have 21,000 genes to code for 100,000 protein, over 100,000 proteins. So it's not like there's a 1 gene to 1 protein ratio thing going on, right? That's not the way it works. Genes code for more than 1 protein. And one of the ways that they do this is by exon shuffling or alternate splicing, sometimes it's called. It basically means we don't have to take those exons. When we take out introns, we don't have to keep the exons in the same order. We can swap them around. And we get different proteins when we rearrange the exons. Maybe you take a particular gene, and one time you take out this intron and this intron, but you leave this other intron in. Or maybe you take this exon and you put it here. Or maybe this intron from one version becomes an exon that's farther up or farther back in a different version. So you can make a lot of different versions by shuffling these exons around and by leaving some introns in, taking some more out, whatever. There's a lot of different ways we can shuffle up that single sequence of RNA to get a variety of different proteins when we go to do translation. So here we are with looking at these exons, and like I said, you can see you can make it with this one or you can make it with this one, and it's a lot more complicated than that. Like I said, you could make it with both. You can make it with both, but with this one on this side. You can make it with this bit over here and this bit over here and this bit. You get the idea. So removing introns is what we call post-transcriptional processing. So it's after we've done transcription, we are processing that mRNA. And so we're taking this primary transcript, which is just copied from the DNA. We're cutting out pieces of it. We also do a couple of other things. First of all, we need to talk about what exonucleases are. I've mentioned this before, but exonucleases digest nucleic acids from the 5' end. So if they have access to a 5' end, they will remove the first nucleotide, the second, the third, and they'll just go right down the line. Nom, 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 nom. These are always present in your cells. That ready to digest RNAs, ready to digest single-stranded DNA. Those are there as an antiviral mechanism. But we don't want those to digest the transcript. We just spent all this time and energy making it. We chose which stretch of DNA we wanted to make an RNA copy of. We removed introns. We did all this work on this thing, and suddenly an exonuclease is going to digest it. We want to prevent that, so we put the little molecule, stick it on the 5' end, and suddenly it doesn't have the shape that an exonuclease can grab a hold of because we stuck an extra, it's really just a modified nucleotide, but we stick this little weird thing on the end of it. We call it a 5' cap. And this 5' cap blocks exonuclease function so that we don't digest this transcript that we just made. If you imagine a cell, you're making protein, you've made RNA, ribosomes use that RNA to make proteins, and we want to stop that. So we turn off the transcription factors, shut off that gene, maybe bundle it up, whatever. We do something so that we're not going to produce that transcript anymore. But we've still got a bunch of those transcripts out there. We just made maybe hundreds or thousands of them, and ribosomes are able to grab them and continue to make proteins. We want that to stop when we turn off the gene. So each transcript actually has an expiration date. It can only be used for a limited amount of time so that they don't hang around continually making proteins that we don't need anymore. That expiration date is called the poly A tail. So we have this 5' cap, like I said, and then we also have this poly A tail. And poly A just means a whole bunch of adenine nucleotides. So A, dozens of them, or maybe a couple of hundred of these A's, just one right after another. And so what's going to happen when we get this out to the ribosome, as ribosomes go through this transcript and use it to make proteins, every time that happens, we're going to shorten that poly A tail. It gets edited. Cut off a little piece, cut off a little piece, cut off a little piece. And eventually it gets so short that tells another enzyme to remove the 5' cap. And so the 5' cap is removed, and now the transcript is digested, and we're not making a protein from 6 months ago when we wanted to express some protein. We are only making the proteins that we are currently making transcripts for. So we've got a little animation here. The PowerPoint that I share with you on Canvas later we'll have a link to this. I don't know if it'll work in slides. But we're looking at the nucleus. We're looking at a piece of DNA. We've got all these transcription factors and activators and things like that. As we go through this, these proteins are all going to, and they're called different things. Don't worry about it. All of it's transcription factors for us. That's all going to function to summon RNA polymerase. It's this big red mitten here, and activate that polymerase so that we can do transcription and produce our messenger RNA. Now there's capping enzymes here, and they're able to pop a 5' cap on that thing's ass. And as we go farther down the line, we're going to assemble our SNRPs, and our SNRPs are going to remove introns. I believe this video may call it a spliceosome. That's a kind of a combination of SNRPs work together to form a spliceosome. Spliceosome is just the body that does the splicing. Clever name. So there's a spliceosome, and that removes our intron in a little loop, and now we've got just exons in the RNA. But notice how that happened while we were doing transcription. Then a poly A tail is added. We stick a whole bunch of A's at the end, and now we've got a mature transcript. So we have to do three things to the RNA to make a mature transcript: 5' cap, poly A tail, and remove introns. And that mature transcript is now ready to leave the nucleus. It's going to be escorted by some proteins. We don't need to worry about them. But we leave the nucleus, and we're going to go on to do translation. And as a little preview, let's see what's going to happen to this transcript. We'll talk more about translation later, but we want to look at what happens to this just so we can see where the poly A tail and the 5' cap come into play. So again, some proteins involved in escorting, and really, even though it's showing several proteins you don't need to know about, it's still only showing a fraction of those that are known. Keeping it simple. This model is more complex than what we really need, but it's also still just a model. So there's some ELVs involved apparently, but this is the small subunit of the ribosome, and it's got a tRNA with it that recognizes the start codon. So we have what's called a start codon. That's the beginning of where we're going to translate. So we have a little bit here that we're not going to translate, right? This part is not going to be translated. So the small subunit of the ribosome is going to summon the large subunit of the ribosome, completing the ribosome, and now we can poop out that protein. That blue guy there is the protein. And this is our stop codon, that UAA, and that's what tells us to stop doing translation. And so we're done. We've made a completed protein here. And if you watch the poly A tail, you can see that it gets shorter every time a ribosome gets there. And when it gets short, we remove the 5' cap, and then exonucleases can digest our RNA. So that's transcription. We'll do translation in the next video. If you have questions about any of this, seriously, shoot me an email. We can very quickly pop open a Google Meet, and we can ask our questions face to face and get your answers. Make sure it all makes sense to you. You don't necessarily have to know the names of everything, but you do need to understand what's happening each step along the way. And so if you have questions, please make sure you ask. You're going to need this, I guarantee you. Hope you guys are doing well. Miss you a lot. Hope to talk to you soon.